dear all students. Today we are here to discuss about artificial vegetative propagation, which is under the unit 5, that is life process of science and technology subject of grade 8. Are you ready to join the class? Okay, all of you, take your stationery and come front. Let's discuss about the artificial vegetative propagation. In the previous class, we discussed about the vegetative propagation, where we have mentioned there are two types of vegetative propagation, that is natural vegetative propagation and artificial vegetative propagation. In the previous class, we have discussed about the various types of natural vegetative propagation. Today, we focus on artificial vegetative propagation. Can you define artificial vegetative propagation? Yes, artificial vegetative propagation means it is human-induced vegetative propagation. It is also a vegetative propagation, but there is an involvement of the human being. It doesn't take naturally, but human made it to happen. So that's why it is called artificial vegetative propagation. Have you seen some people's growing the plants in a different way? Some used to cut the small piece of root, stem and leaves and plant a new plant. And some used to just join. Have you seen sometime the people used to cut a small piece of stem of one plant to the another plant and grow a complete plant? Have you seen? See here same thing is doing. That is called grafting. And somewhere we can see like lemon. A small branch of lemon is just kept under the ground, and when the roots arise from it, it will separate it from that and it will grow into a new plant. And similarly, there are many advanced techniques are developed, especially in a sophisticated lab. If there are some endangered plant which are not able to grow into a new plant, and we have to preserve those plants for the future generation, what the people do? Now there are modern technology in the laboratory. They take the small pieces of the tissue from those endangered plant and they used to grow in a artificial cultural media. Then they grow many plantlets of the same plant. That is called tissue culture. So there are various methods of artificial vegetative propagation, and it is an advanced plant reproduction technique. That means there are different advanced techniques are used, so it is also known as advanced plant reproduction technique. Now, what is the exact definition of the artificial vegetative propagation? Yes, there is production of new plants asexually using tools and techniques. Okay, simply we don't define this artificial, just like in vegetative propagation, producing the new plants from vegetative parts we described, yeah. But in case of artificial vegetative propagation, there is a using tools and techniques. We are growing the new plants from stem, root and leaves, that is vegetative parts. That is called artificial vegetative propagation. What is the purpose of it? The purpose of this artificial vegetative propagation is improvement of the quality and quantity of the crops or cash crop. That means we can get cash from those crops. Okay, just we can improve the economic condition of the family, even in the country also. That means if we improve the quality and quantity of the crops, we can, what we can do? We can export to the different countries and we can get the cash also. That means cash crop we can say it. And next one is, what are the key benefits of the artificial vegetative propagation? Can you see this picture? See, in a small pot also. This is a potted plant where many oranges are fruiting. Yes, there are many fruits on the same small plant. How it is possible? If we see, that means sow the seeds in the ground, it takes long time to grow, and when the plant become very big, then only it fruit, that means it develops fruits. But here in a small plant which is grown in a small pot also produces many fruits at a time. This is possible because of this artificial vegetative propagation. And it copy the exact genetic copies of the parents. How the parent is, the new plants will be same, just Xerox copy of their parents. And they can yield fruits earlier than the seeds grown plant. That means the plants which are grown from the seed produce the yield the fruits later, but these vegetative propagation plants yield the fruits very earlier. Next one is common methods of artificial vegetative propagation. What are the common methods which are used in the artificial vegetative propagation? There are cutting. Simply cutting. Cutting means either cutting the root, stem or leaf, and from that twig we can grow a new plant. That is simple cutting. Next one is layering. Just lying down some flexible part of the plant into the soil, then growing a new plant. That is layering. And another one is, this is also a kind of layering, air layering it is also called, and that is also called goti. Here we are growing the roots on the air. Aerial part of the plant is tied with mud and dung mixture, then that will grow roots, and this will be transferred to the next place and it will grow into a new plant. Another one is grafting. Here in the grafting, what we do, we take one part from the one plant and we just join to the year-old plant having root, and these two are tied together and it will form into a new plant. Okay, this type of vegetative propagation is called grafting. And tissue culture, where it is a very advanced technology where there is used, it is a advanced techniques where we can grow a large number of plantlets from a small piece of tissue. This is called tissue culture. We discussed about these 5 types of artificial vegetative propagation today. Okay, first one is cutting, a simple propagation method, most easiest method of vegetative propagation. See, this may be by cutting the aerial stem, or by cutting the underground stem, or by cutting the roots also, or by leaves cutting also. One piece of leaf can be cut into small pieces and it is dipped into water for some time and it will grow the roots on its midrib region, then it can be separated into a new plant. In the similar way, in case of stem also, just we take a small twig of the stem which is kept in a water for some time, some rooting hormone is placed and due to that roots will develop into it, then new plant will be grown. This is the method of propagating through the cutting. Here what is cutting means? Sowing cut piece of leaf, stem or root to grow new sapling, that is called cutting. Here we use year-old twigs with at least 2 nodes. There should be node. If there is no node, already I have discussed in a previous class also. In case of vegetative propagation also, we discussed about the node and internode. From the node region only, what happens? The roots will develop. Just in the previous class also we discussed about the node and internodes. Yeah, this is the node region which we, while cutting also, see, if we are doing the cutting method also, at least 2 nodes should be there like this, and it should be dipped into the water. From this node region roots will grow and shoot system will also grow, and that will be grow into a new plant. And next, while choosing this cutting, we should choose the twig which should be a year old, okay, and leaf also somewhat mature leaf, roots also mature leaves, mature roots, so that it can grow a new plant easily. When we grow the new plants from the cutting, that is the easiest way of the artificial vegetative propagation. Other artificial vegetative propagation somewhat complex than this cutting. Examples of cutting you can see here: root cutting. From the root also we can develop like raspberry, ground raspberry, blackberry. These are grown from root cutting. This is a raspberry plant which is growing from a root piece. And next one is stem cutting, like sugarcane, rose, pear, lemon, etc. can be grown from the cut of the stem. And leaf cutting, like snake plant, we can cut the part, that means one piece of snake plant leaf that can be grown into a new plant. These are the different examples of cutting. Next one is layering. What is layering? Layering means bending branches of new, that means bending branches for a new growth of the plant. Just simply, if we have a plant, suppose there is a plant which have many small branches, just what we do, this branch will be just kept down and it will be just, this is soil, under the soil we keep. After some time this will grow roots and we cut from this part and we separate this new plant from this parent plant and it will act as a new plant. In this way we can develop the layering, or by the process of layering we can develop new plant. This method is called layering. Okay, this layering is suitable for the plants with soft and low branches. That means the branches which are soft or flexible branches and which used to grow lower part, okay, in the low level ground. Those which are in the upper side we can't just bend in and keep inside the soil. If they are growing in the lower part, we can easily bend and just bury under the soil to develop the roots. Here what we do in the layering process, first of all we select a branch near the ground, which is nearer to the ground, we select it. Then just we remove 2-3 centimeter of its bark. Then we just bend and bury the branch in the soil, in the, just like here. Okay, one branch is taken, its some part 2 to 3 centimeter bark is peel away, then it is buried under the soil. After some days there are roots and also a small branch or a small shoot is developed. If we cut from this place, then we can get a new plant. In this process we have to keep the soil moist until the new roots or the shoot is developed, and after developing those roots and shoot we can separate it and we can place in the different place. There are layering process is this, but the methods of layering may be different. There are different methods of layering. First one is tip layering. See, in case of tip layering, what we do, a tip of the plant is buried under the soil, and from that tip roots and shoots are developed. And another one is mound layering. In case of mound layering, we don't bend the branches, we let the branches how they are, just we keep the soil above them, just we bury the that means the later part or the lower part of the branches through the soil. Just we bury with the help of soil, and this is called mound layering. From each branch we can separate the new plant. That means when we bury these branches through the soil, all will grow the roots, and we separate from the parent plant and we develop the new plant. Next one is simple layering. In case of simple layering, just I have explained here in the board also, one branch is lowered and buried under the soil. When the roots comes, then we separate it from the parent plant. And another one is air layering. It is also called goti. In case of air layering, the aerial branch is selected and some part of it is, that means bark is removed, and with the help of mixture of, that means soil and dung, is just wrapped around it with the help of plastic. Then when the roots comes out, then we cut it and separate it from the parent plant and we get the new plant. Next one is compound layering. In case of compound layering, what we do, a same branch is just bent and buried multiple times in the soil, and a single branch, that means from the single branch we can get many new plants. This is called compound layering. In this way we can get different plants or the many plants from the different methods of layering. Now just discuss about the air layering or goti. What does it mean? Creating roots on the branches while still attached to the parent plants in the air. As it is in the aerial part, it is called air layering also. Just like this. Already we have discussed about it. And what type of plant does it suit for this goti or the air layering? Lychee, mango, guava, orange, pomelo, etc. follow this method of artificial vegetative propagation. How it is done? What is the process of it? See, simply, in the first step, what they do, they remove the bark, 2 to 3 centimeter bark from year-old branch, 1 year old branch. Then after removing that, what they do, cover with the nutrient-rich soil and plant hormones. That means in that especially we mix soil, dung and some plant hormones like that. That means root hormones are specially placed in it. Because of that, it can produce the roots, and it is tied with the help of plastic that prevent the infection on the plant. Then we wait for 1 to 2 months for root formation. Then we, after growing the roots, we cut it from here and we grow into a new plant. In this way, we can develop a new plant. This is called air layering or goti. Next, and this is the process which is mostly followed in the farmers. That means most of the farmers used to follow this method to develop the new plant from the lemon, orange, apple, etc. Next one is grafting. Grafting, it means joining plant parts. One plant shoot is connected to the another plant root system mainly. See here, what is the definition of grafting? The process of joining two parts of the closely related plants. Here you can see there is only one shoot is connected to the another plant. That means the plant which is taken as a shoot, that is called scion, and that with root system, that is called stalk. Here this down one is stalk, it has the root in under the ground, and that upper part which is taken from the another plant, that is called scion. In this picture we can see there are two scions are kept in the same stalk so that we can grow the new plant. Mostly when we do this, they should be closely related. That means genetically they should be related with each other. Otherwise we can't grow. For example, if we want to grow citrus plant and apple plant together, it is not possible. But citrus family, different plant like lemon, orange, and can be grafted. Okay, and like tomato and potato we can, because they are from that belongs to same family, similar. And brinjal and tomato, bromato, have you heard? This can be grown. So these are the method of, that means grafting. In case of this grafting, what they do, first of all, joining two parts of the closely related plants, and what is the purpose of it? Prepare plantlets of the fruit plants with good productivity. In this case, while selecting the scion and stalk, what they do, the scion of the quality fruits or the good quality plant scion is selected, and the stalk may be, that means simply a simple plant stalk can be chosen. For example, if we grow a large mangoes which give more product, simply what they do, the branch of the mango which gives large fruit and many fruits at a time, that branch is selected as a scion. And the year-old new plant which is growing somewhere, that means that is not a that means a very qualitative mango, and just they cut the upper part of the year-old twig, and that branch of the quality mango is connected to that, so that they will wrap it tightly, and after some time they can get a complete plant which can form the, that means in a faster way they will develop the large mango fruits with quality, and at a time that can produce many mangoes too. The main here key components, what are those? Stalk, rooted part from a year-old seedling, and scion means twig from a selected high quality plant, just I explained. And how this grafting process is done, just see here. First, we prepare the scion and stalk. Already I have told you, scion is always selected from the branches of the already grown fruiting plants. Already it has produced the fruits. And stalk is just a year-old plant which is growing from a seedling. Okay, and then what they do, the cut and shape join points for the proposed to fit. For example, it can be, sometime we can, there are different shapes we can cut, like in some cases, suppose this is a stalk, okay, and in this stalk we can see cut it into V shape, okay, and for that the scion should be of, that should be fixed in this part, okay. In this we see, if there is a V shape, it should be pointed like this, so that it can be fixed here. And after that, another method is, if we are cutting in this way, suppose this is slanted one, and the scion should be cut in the same way in the slanted shape, that's why it will be fixed together. Because of that, there is, that means there is conduction of water, mineral, those conductive tissues should fix together, that's why it can conduct the water and mineral from the soil. Okay, the shape should be cut, scion and stalk shape should be cut in such a way that should fix together. While that means when we bind with the plastic or the tape, it should, the conduction of water, minerals should be continued through those conducting tissues. And so here we should ensure that internal conductivity between the stalk and scion should be continued when we after join it, and join parts using grafting wax and plant tape. That means to fix them properly, we should use the grafting wax and plant tape, so that we can tie it tightly, just like here. See, here it is tied with the tape, okay, and allow time for healing and firm joining. It doesn't fix in a very short duration. To fix it, it takes time. So we have to allow the time to form the exact joining, and they form a complete plant. That means it will result into a single plant with science quality characteristics. That means even though we are using a stock of year-old plant which is growing from a seed, as we are selecting the scion from the good quality plant, that means which we have the desired character on it. That means what are the desired characters we chosen from that plant, it carries from the scion. That's why the newly formed grafted plant will have the same quality of that scion. Now next one is tissue culture. Another method of artificial vegetative propagation is tissue culture. In this tissue culture, these are advanced propagation techniques than other. Here, simple layman or the simple farmer cannot do tissue culture. It needs a advanced technology where there is a manpower also advanced way. That means who has the knowledge about the biotechnology, they can perform this. But nowadays farmers are also given training about these. That means it should be carried out in a sophisticated lab where there is incubator, all those things. That means all the laboratory equipment which are required for it. The definition of this tissue culture is producing plantlets from the small pieces of plant tissue in a laboratory. Okay, that means we produce the large number of plantlets from a simple single tissue of the plant, either from its root, stem, leaf, fruit, flowers, any part. From that tissue, we can grow many new plants in the laboratories by using the artificial media, so that we can grow the new plants in a very short period of time. What are the benefits of this tissue culture? Large scale production of disease free plantlets. At a time we can produce large number of plantlets or the plants which are disease free. And genetically desirable traits. What kind of traits we are desiring, those characters can be chosen. And there is rapid multiplication. That means in a short period of time we can multiply the number of plants. So that's why it is a rapid multiplication of the plants. Now how this tissue culture is done? Tissue culture is especially done in a sophisticated lab with a highly technical person. First of all, we select and prepare explant. That means explant means small piece of plant tissue. Already I have told you, any parts of the plant. Then see, from this we can take a piece of leaf, stem, anything. Then we cross it into small pieces. That means that tissue also crossed into just cells. Then these explants are kept in the cultural media with required nutrients. These are the artificial media prepared in the laboratory, which contain all the plant hormones, nutrients which are required for the growth of the plants. Then at that time a mass of tissue is developed here. That mass of tissue is called callus, which is undifferentiated mass of tissue. Now this callus is transferred to the plant hormones. That means that in that plant hormones, that callus will be separated into small pieces and it will placed in another container where it contains plant hormones. That means that can grow roots and shoot. Like auxin hormone and cytokinin and auxin hormones are placed in the media, then it will grow the roots and shoot on it. Small plantlets will be developed. Those small plantlets will be differentiated into small pieces. That means separated from that callus. Okay, one plantlets. And then those are transferred into the soil, or it is also called acclimatization. That process is called acclimatization. And those are transferred into, that means cutting into small pieces and separating into the different part, then it is that small plantlets will grow. This whole process is called acclimatization, and those small leaflet, that means plantlets, which are now able to grow, that means they are grown in a nursery separately for some time, that means for a year long. Then after that, when they are able to adjust in a outside environment, then that will be transferred to the different places. And in this process, it produces genetically identical plants, all the clones. See here, these are the plants which are grown from the tissue culture separately in a soil. They see all are identical, and these are, that means some of these are the plants which are grown from the tissue culture. Okay, and this is also advantages of the vegetative propagation. At a time, we can produce many identical plants from a small piece of tissue. And these are another advantage of the vegetative propagation is faster than growing from the seeds. All the vegetative parts are, that means that grow very faster, and they can yield the fruits also very faster, and preserves the desirable traits. Whatever the traits we required, that we desired, those traits or the characteristics can be continue in the next generation. But in case of sexual reproduction, that means if they are grown from the seeds, we can't maintain the desirable traits in those plants. During sexual reproduction, there is crossing over takes place, that means the mixing of parental characters takes place. Because of that, we can't maintain the desirable traits. But in this vegetative propagation, whatever the traits we want to continue in the next generation, we can continue in those next generation plants. Some plants rarely produce viable seeds. That means some plants rarely produce viable seeds, like potato. We can't grow the potatoes from the potato seeds. The seeds of the potatoes are not viable. At that time, what method we should follow? That is vegetative propagation. Can produce plants year round. That means when the plants reproduce or produce from the seeds, they may be the seasonal plant. But by using these methods, artificial methods or artificial vegetative propagation or natural vegetative propagation also, we can produce the plants year around. That means in the on season also, off season also, we can produce fruits. Next one is advantages of artificial vegetative propagation. Especially from the artificial vegetative propagation, what are the advantages? See here, this is directed breeding and genetic as per the need. We can breed it, that means it is a directed breeding, where we maintain the genetic characteristics as per our need. And it produces the fruits very faster. In the artificial, that means like cutting, grafting, when we do cutting, grafting, etc., we choose the plant which is already fruiting. So it can produce the fruits very faster than the plants which are growing from the seeds. And rapid production of genetically identical sapling. At a time, we can produce so many saplings or the small plantlets. And useful for the plants without viable seeds. This is also very important, and these are the advantages. Now, even though there are so many advantages from the vegetative propagation, there are some disadvantages of the vegetative propagation too. Like it is limited genetic diversity. We can't find out the genetical diversity there because we are just maintaining the copy of the parent in the offspring. And may spread disease easily. It can spray. If the parent plant is diseased, then all the plants, that means, will be diseased. And some methods require specialized skills or the equipment. Like in case of tissue culture, the only technician can do that, other person cannot do. Not suitable for all plant species. It is not suitable for the all kinds of plant. It can be more expensive than seed propagation. Sometimes it may be more expensive than like tissue culture may be expensive than the seed culture. These are the disadvantages of the vegetative propagation. We have discussed about the various types of artificial vegetative propagation. Let's evaluate ourselves through certain questions. Okay, first question is, which method of artificial vegetative propagation involves joining two parts of closely related plant? Can you guess? Cutting, layering, grafting, tissue culture. Which one is correct option? Yes, it is grafting. Okay, next question. In the grafting process, the part with root obtained from a year-old fruit plant is called what? Stalk, scion, graft, layer. Which one is the correct answer? Stalk. Very good. And next one is, what type of vegetative propagation is shown in the given experiment? Here one experiment is given in the picture. What kind of vegetative propagation is shown here? Grafting, tissue culture, air layering, cutting. Which one is the correct option? Okay, very good. This is tissue culture. And next one is, what type of vegetative propagation should be adopted to grow a plant producing potato, tomatoes in a same plant as shown in the figure? See, in the soil there are potatoes, in the upper part, in the shoot system there is tomatoes. What techniques can be applied to grow such a plant? Okay, tissue culture, grafting, air layering, cutting. Which one is the correct option? Yes, it is grafting. Next, some short responses questions. Write the difference between layering and Goti method of the propagation. Here layering means simple layering and Goti means air layering. Okay, you can write the difference between them. And why is artificial vegetative propagation preferred for plants without viable seeds? That means why it is important to use artificial vegetative propagation? For those plants which cannot produce viable seeds. That means viable seeds can produce the new plant. But if the seeds are not viable, they cannot produce the new plant. So, why it is important? You can explain based on our discussion. What is the purpose of using grafting wax and the plant tape in the grafting process? We discussed in the class. Please watch the class or revise the class and write the answer. Next, last question. Compare and contrast the advantages and disadvantages of artificial vegetative propagation methods versus growing plants from the seeds based on genetic diversity, time of maturity and resource requirements. Based on these three characteristics, please compare and contrast between the that means advantages and disadvantages of artificial vegetative propagation methods versus seed growing plants. We have discussed many things about the artificial vegetative propagation. We discussed some questions also. I hope you have all understood the different artificial vegetative propagation and you will apply in your garden. For today, bye bye. See you again.